My name is Dr. Mehal. I'm an investigator and a liver specialist at the Veterans Hospital in West Haven, Connecticut, and at Yale University. My group is interested in the liver condition NASH, which is the most common liver disease in the United States. It is particularly prevalent in our veteran population and is well known to be associated with obesity and the metabolic syndrome. It is, however, not known exactly how obesity results in the development of NASH. This study that I'm about to present to you will ask this question, and specifically ask the question whether there is an increase in the ligands for a TLR known as TLR9 in patients with NASH, and whether this is responsible for the development of NASH in patients with obesity. Our hypothesis for this work was that mitochondria inside hepatocytes are at risk of oxidative stress, and that with a high-fat diet that is prevalent in developed countries, that this results in oxidation of DNA. Oxidized DNA is known to be a ligand for TLR9, and release of such DNA into the circulation would allow it to be accessed by TLR9 on macrophages and Kupffer cells inside the liver. Subsequent to this, activation of TLR9 would result in production of inflammatory cytokines such as IL-1 beta and TNF-alpha, which would result in steatosis and inflammatory injury on hepatocytes. This can be blocked by the use of a TLR9 antagonist, which should be able to stop the development of NASH and might also be able to reverse NASH once it has developed. This study was done by testing plasma from 3 groups of subjects. One group of subjects were lean, a second group of subjects were obese but had no evidence of liver injury, and a third group of subjects were obese with evidence of liver injury. In addition, we used genetically altered mice and also used a molecule which is known to be an antagonist for TLR9 to see if we could induce a halt in the development of NASH. We examined plasma from 3 populations. Population 1 consisted of individuals who were lean. Population 2 consisted of individuals who were obese but with no evidence of liver injury. And population 3 consisted of individuals who were obese with evidence of liver injury. As can be seen, the level of mitochondrial DNA was significantly higher in population 3. This was in contrast to the level of nuclear DNA, which was consistently the same in all 3 populations. We were very interested in the form that this mitochondrial DNA was taking in the plasma. As can be seen from this flow cytometry plot, on the x-axis you have staining for plasma membranes, and on the y-axis you have staining for intact mitochondria. So on the top left-hand quadrant, one sees intact mitochondria in the plasma. In the bottom right-hand quadrant, one sees microparticles alone, and in the top right-hand quadrant, one sees mitochondria within mitoparticles. And there was an increase in the number of mitochondria within microparticles within group 3. We also wanted to identify the source of these microparticles, and as can be seen from the Western blot, when blotted with a marker for hepatocyte plasma membrane, arginase 1, there was a strong signal. However, there was no signal for platelet plasma membrane markers, which are the other known source of microparticles containing mitochondria. We also were very interested to see if there were differences in oxidation status, and as can be seen, using an antibody which will detect oxidized DNA, population 3 had a higher percentage of oxidized DNA within its mitochondrial DNA population. And finally, to test whether the microparticles were actually relevant for activation of TLR9, plasma from individuals was used to activate a TLR9 reporter cell line with and after removal of microparticles, and removal of microparticles resulted in a significant decrease in the ability of the plasma to activate TLR9. Subsequently, we wanted to test if the mitochondrial DNA from livers which had been exposed to high-fat diet had more pro-inflammatory capacity. And as this figure shows, if mitochondrial DNA is removed from hepatocytes that came from mice that were either fed a controlled diet or a high-fat diet, there was stimulation of TNF-alpha production on primary mouse Kupffer cells. However, if the mitochondrial DNA came from mice that had been fed from a high-fat diet, the amount of TNF-alpha produced was significantly higher. In contrast to this, the amount of interferon alpha and IL-1 receptor antagonists produced was no different. What this does is it shows us that mitochondrial DNA from hepatocytes from mice fed a high-fat diet have the capacity of increasing pro-inflammatory cytokine production, but not necessarily production of a whole range of other immunological molecules. We also wanted to see if Kupffer cells from mice that had been on a high-fat diet had different responsiveness to TLR ligands. And what this lower panels show is that in response to CpG, which is a TLR9 ligand, that Kupffer cells from mice on a high-fat diet were increasingly responsive as compared to Kupffer cells from mice on a control diet in terms of production of IL-1 beta, IL-6, and TNF-alpha. To try and identify the cellular requirement for TLR9, we used a Cree-Flock system in which TLR9 was removed from lysozyme-expressing cells. The next figure shows that wild-type mice that are on a high-fat diet have significant steatosis and inflammation as expected, and mice that are entirely lacking TLR9 have a significant reduction in these inflammatory markers and steatosis as previously shown for other models of NASH. Significantly, mice in which TLR9 had been removed only from lysozyme-expressing cells, predominantly Kupffer cells, there was also significant protection from NASH, and this was recapitulated in the levels of ALTs, which was significantly less in the TLR9 Kupffer cell-specific deficient mice. Finally, we wish to see if a TLR9 antagonist for which safety data is available could provide some protection. And this was done in mice that were wild type, and again using a high-fat model of NASH. And if a TLR9 antagonist was given starting 8 weeks into the high-fat diet, there was still significant reduction in steatosis and inflammation, and also in transaminitis. This work was possible through a collaboration between 3 academic centers and Dynavax. It really highlights the importance and helps us focus on mitochondria inside hepatocytes as a key component of the development of NASH. It also demonstrates that the difference between individuals with obesity with and without NASH might actually be predominantly in the absence and presence of TLR9 ligands, and further identifies the ligands and TLR9 as a therapeutic target for NASH. We are grateful to our funding sources from the Veterans Office of Research and Development, from the National Institutes of Alcohol Abuse and Alcoholism at the NIH, and also the National Institute of Diabetes and Digestive and Kidney Diseases at the NIH.